All right, let's jump right in. We're going to talk about a technology that is, right now, quietly redesigning the very foundation of our food supply. You can think of it like a genomic crystal ball, something that lets plant breeders literally predict the future, supercharging our ability to grow better, more resilient crops. And this is not some far-off hypothetical question. It's one of the biggest challenges of our time. The global population is booming, and so is the demand for food. But at the same time, our crop productivity is getting squeezed by all the stresses of a changing climate. So the real question is, how on earth do we keep up? To really understand where we're going, it helps to look back. Take a look at this number, 775 million tons. That was the global wheat harvest in 2023. Now, what's absolutely incredible is that we grew all of that on pretty much the same amount of land we used back in 1961, when the harvest was less than a third of that. That huge leap was thanks to advancements in plant breeding, and it just goes to show what's possible when we get innovative. So here's how we're going to break it all down. We'll start with why we need this stuff in the first place, then look at how breeding has evolved. Then we'll get into the main event, this genomic crystal ball. We'll see how it works, how we can make it better, and how it's already changing our food. And then finally, we'll take a peek at what's coming next. For, centuries, breeding was really a game of patience and just observation. Breeders would walk the fields, pick the best-looking plants, the ones that were the biggest or seemed the healthiest, and then save their seeds. But today, we've moved way beyond just looking at the plant. We're now looking deep inside it. So this whole evolution kicked off with conventional breeding, which was based purely on what you could see. Then we got a bit smarter with marker-assisted selection, or MAS, that used a few DNA markers to hunt for major genes. It was a great first step for sure, but it was limited. It missed the thousands of other little genes that all work together on complex stuff like yield. And that brings us to today's absolute game changer, genomic selection, or GS, which looks at the entire genetic picture. And yeah, that's what we're really here to talk about. Because this isn't just another small upgrade, it's a completely different way of thinking. We're moving from a process of simply observing and selecting to one of pure prediction. We're literally building a genomic crystal ball. So what is genomic selection exactly? Well, it's a method that uses a plant's entire genetic profile, its whole genome, to predict how it's going to perform out in the real world. We can estimate its future yield, its tolerance to drought, or its resistance to disease, all from its DNA, often before we even bother planting it. Here's a great way to think about it. How does Netflix know exactly what movies you're going to want to watch? Well, it crunches a ton of data, what you've watched, what other people like you have watched, to predict what you're like next. Genomic selection does basically the same thing, but for plants. It analyzes the DNA and real-world performance of thousands of plants to build a model that can predict and recommend the blockbuster crops of the future. It all works in 3 basic steps. First, you train the model. You take a big group of plants called a training population, and for each one, you get 2 key pieces of info, their complete DNA and how they actually performed in the field. Second, you use that data to build a predictive model. Basically, a smart algorithm learns all the complex links between genetic patterns and good traits. And third, you select. Now you can take a brand new seedling, scan its DNA, and use your model to predict its future, all without ever having to grow it to maturity. And this visual here shows that process perfectly. You've got your training set, or TRS, on the left. For these plants, you have both their genes and their real-world performance data. That's what you use to build the model. Then you have a new breeding set, or BS. For this group, all you need is their DNA. You feed that into the model, and boom, it spits out a prediction of how good it's going to be. It's a massive, incredibly powerful shortcut. Okay, but this crystal ball isn't powered by magic. Its accuracy, how well it actually predicts the future, depends on a few really key factors, and breeders are constantly working to sharpen the focus of these predictions. To get a really sharp prediction, you need all the right ingredients. It starts with how you design that training population. Then you need super high-quality genetic data. The trait you're looking for matters too. Some are just easier to predict. You also need really precise real-world data, which is a process called phenotyping. And finally, you need some seriously powerful statistical models to piece it all together. Now, when it comes to that training set, you might think, well, bigger is always better, right? But it's actually a trade-off. A huge set can give you great accuracy, but it's also really expensive, and after a certain point, you don't get much more bang for your buck. So the real goal is an optimized training set, one that's just big enough to be accurate without wasting a bunch of time and money. And here's another really crucial point. Kinship is everything. The predictive model works best when the training plants are genetically related to the new plants you're trying to predict. So, a model trained on wheat is fantastic at predicting new types of wheat. But could you use that same model to predict the performance of corn or rice? No, it just wouldn't work. The closer the family, the clearer the prediction. So, that's the theory. But is this stuff actually working out in the real world? Let's take a look at how this technology is making a real impact, turning genetic code into much, much better crops. Our first showcase comes from CIMMYT, the International Maize and Wheat Improvement Center. They've been using genomic selection to seriously speed up maize breeding in Africa, which, as you know, is a critical food source for the entire continent. Their whole goal is to cycle through generations super fast, picking the winners based on their predicted genetic merit. And the results? They're really impressive. They've seen an average genetic gain of 0.134 tons per hectare for grain yield in every single breeding cycle. That's a real, tangible improvement in what that crop can do, and it's happening so much faster than ever before. Okay, our second example comes from a private company in Sweden, Lantmännen. They're using a really cool, innovative strategy for crops like wheat and barley, which just shows this technology isn't only for big public research groups. And this right here really shows the power of what they're doing. A conventional breeding cycle can take anywhere from 5 to 8 years because you have to wait for all the field trials to pick the best parent plants. But with their genomic selection strategy, they pick the parents super early based only on their DNA predictions. And get this, it cuts the breeding cycle down to about 2 years. That is a massive acceleration in crop improvement. So, where does this all go from here? Well, genomic selection isn't just a standalone tool. It's a huge part of a bigger technological revolution happening in agriculture, one where biology is merging with big data and AI. As one of the sources for this explainer puts it, the integration of this tech with machine learning and AI is a really promising new frontier. Think about it. As our datasets get bigger and our algorithms get smarter, that predictive power is only going to grow. We're moving toward an era where we can design the crops we need with unbelievable precision. And in the end, this all brings us right back to that first big challenge we talked about. Genomic selection is an absolutely key tool for building a more resilient food future. It can help us get higher yields, develop stronger resistance to pests and diseases, and breed crops that are way better adapted to our changing climate, all things that are totally essential for more sustainable farming. And that leaves us with one last big question to think about. As these worlds of data science and biology continue to fuse together, and as our ability to predict the future from a strand of DNA gets more and more powerful, are we on the cusp of a brand new agricultural revolution? One that's driven not by what we can see, but by what we can predict.